Well, about 7 or 8 years ago, we got the first understanding of the presence of bacterial biofilms in chronic wounds through the pioneering work of Garth James and Randy Walcott. And what they did was to look at a series of biopsies from chronic wound patients using scanning and light microscopy. And what they basically found was they could identify structures that were consistent with biofilms in about 60% of the chronic wounds. However, using more advanced techniques, we now know from multiple clinical studies, both from my lab and others, that there are probably over 90% of truly chronic wounds have some component of a bacterial biofilm within their wound bed when they present to you as a truly non-healing chronic wound. And building on Andy's concept of the critical colonization that occurs within this spectrum of wound bioburden, I think it's probably accurate to say that the term critical colonization was an attempt to understand what was critical about the bacteria that were present, but not at elevated enough levels to classify as truly spreading or systemic infection. And I'll propose to you that another euphemism for the term critical colonization is the presence of persistent bacterial biofilms. A nice translation of a lot of the biochemistry and microbiology that I'll talk to you about today is this Biofilms Made Easy. It's a free download from Wounds International webpage. So this is a key question because we are frequently asked, does formation of a biofilm in a wound actually retard healing, or is it simply just there? What's the evidence that it actually is a cause of the wound to not heal? And the answer is, in our animal models, definitely the formation of a biofilm impairs healing. So why do I say that? Well, looking at data from Shirl, who is in Tom Musto's labs at Northwestern University, did one of the best definitive experiments to show that the presence of a biofilm impairs healing. So what they did was to take normal mice, they created full thickness wounds in their back, and they inoculated them with wild type Staph aureus or Staph epidermidis that can produce a biofilm. And they looked at the healing compared to non-inoculated wounds. And as you can see in these photomicrographs, when the wounds are not inoculated, the wounds heal very normally. The level of bacteria at that contamination colonization level is not sufficient to overwhelm the immune system of the mice, and they heal normally. But when there is a huge inoculum of Staph aureus or epidermidis, enough that some of the planktonic bacteria can now attach and convert into biofilms, now there is a very significant and reproducible impairment of wound healing due to the biofilm. Well, now you're probably appropriately saying, look, that biofilm is there, but prove that it is the biofilm that's actually causing that impairment. And so to answer that question very directly, they used an inhibitor of the quorum sensing system that tells planktonic bacteria to convert into biofilm. Everything else about those bacteria is exactly the same. And when they blocked the formation of the biofilm phenotype with the quorum sensing inhibitors, now there's no difference in the level of wound healing, even if they inoculated those wounds with 10 to the 5th colony-forming units of the wild-type bacteria. So this is some of the best data that proves that in these animal models, the presence of the biofilm is what is impairing the healing. And basically what happens is it overwhelms the immune system and enough bacteria persist to produce the biofilm. So why does the bacteria impair healing when it's in the biofilm? Well, again, pioneering work from Bill Costerton, who coined the term biofilms, showed that when planktonic bacteria, the white ovals here, contaminate a wound, that are in our normal immune system, both our phagocytic macrophages and neutrophils, as well as our antibodies, are able to effectively remove and kill those bacteria. And if we want to add antibiotics, they kill very well too. But if some of these planktonic bacteria survive long enough to attach to a surface, either a orthopedic implant or a piece of collagen in the wound matrix, they quorum sense and will eventually convert into a biofilm. Now, both our antibodies and our antibiotics can't penetrate well to that biofilm. And in addition, the inflammatory cells can't engulf and kill them because the biofilm structure is many times bigger than they are. And what happens is that the inflammatory cells still activate, and they secrete the proteases and reactive oxygen species that they normally use to kill planktonic bacteria. But instead of being able to effectively kill the biofilm, the biofilm persists, but these proteases and reactive oxygen species have off-target effects that begin to damage the surrounding tissue. So I'm going to show you just one slide that I hope will indicate to you how critical the level of the proteases are in a wound. So if we look at a chronic wound and we measure its area, shown in the red squares, over a period of about 90 days with weekly measurements, and we also simultaneously measure the level of protease activities in wound fluid taken at those same times, you can see that as this patient comes in and starts getting good wound care, the wound area decreases. The wound is healing. The protease levels are also dropping. But then this patient had what you frequently see is the wound stalls. It does not continue on this progression, and it just flattens out for about the next 6 to 7 weeks. Well, what's happening at that time is that the protease activity levels are going up. This wound is developing a biofilm. And what happens only after appropriate debridement and treatment with topical antimicrobials do the protease levels begin to drop, and then the wound can enter back into the healing phase and come out of that stall phase. So another important question for us to ask in terms of how you do biofilm-based wound care is, why are bacteria so hard to kill? Well, there are multiple answers. One of the most important is that the exopolymeric matrix of the biofilm is a very dense matrix. I'll show you some scanning ends. It's very difficult for large molecules to easily penetrate that, such as our own antibodies. In addition, many of the chemically reactive antiseptics, such as hypochlorous acid, actually get neutralized by reacting with the components of the exopolymeric matrix. And finally, the components of the exopolymeric matrix are usually highly negatively charged through the negatively charged polymer sugars or through the presence of bacterial DNA. And this negative cloud actually tends to ionically bind positively charged ions such as silver, many of our antibiotics, and in some cases PHMB. Another issue that I'll show you is that there are persister bacteria in the center of these large biofilm masses. And if there's one thing I want you to remember from my presentation is that antibiotics only kill metabolically active bacteria. They inhibit enzyme systems in the bacteria, so if the bacteria are not metabolizing, antibiotics are almost totally ineffective. In addition, the center of the biofilm becomes very hypoxic because only the outer shell of the biofilm are metabolically active bacteria, so that promotes a special niche, a special area within the biofilm center that is very, very low in oxygen, allowing strict anaerobic bacteria to grow. And then finally, sort of the sum of all fears is when we find within the polymicrobial biofilms in wounds that there are multiple species, including frequently resistant bacteria such as MRSA. And they provide bystander effects by secreting enzymes or other molecules that inactivate the antibiotics, so that a sensitive Staph aureus next to a MRSA actually provides benefit from being that physically close to the MRSA bacteria within the wound bed. Well, let me just give you two quick examples of how tolerant bacteria become to antibiotics when they are in a biofilm. So again, working through the work of Phil Stewart and others at Montana State, they grew Pseudomonas aeruginosa in the planktonic culture form, and they exposed this high concentration of 10 to the 6th colonies of Pseudomonas to 50 times the minimum inhibitory concentration of tobramycin. And after 4 hours, you can see it killed every bacteria present. That's what antibiotics do. They kill metabolically active growing bacteria. So if you took those same sensitive bacteria and grew them in a mature biofilm and exposed them to over 100 hours, 4 days of that same concentration of antibiotic that killed every planktonic bacteria in 4 hours, after 4 days it's killed less than 1 log. So this shows you that a sensitive bacteria becomes tolerant to the antibiotics when it is in the presence of a biofilm. Why does that happen? Well, it's because most of the bacteria within the center of the biofilm mass are not metabolically active. They're so-called tolerant or dormant persister bacteria. And using a Pseudomonas that turns green only when it is metabolically active, you can see in this data, again from Phil Stewart's group, that only the shell of the biofilm is metabolically active, the center is inactive, and for the second time, antibiotics only kill metabolically active bacteria. Well, what about antiseptics? Again, from data from Bill Costerton's group, Bill grew Pseudomonas on an inert surface and exposed it to 1 hour of quarter-strength Dakin solution and then used a dead live stain. And you can see the outer layer of the biofilm is killed. But when you look at the center of the biofilm mass, they're still alive. Why does that happen? Because the bleach is so chemically reactive that it is neutralizing itself by chemically reacting with the outer shell. And so the penetration, even after 90 minutes of the bleach into the biofilm mass, only has penetrated about half the distance. And this is what has been called the reaction diffusion problem. Chemically reactive antiseptics tend to get neutralized by reacting before they can diffuse. So the good news is that the principles of biofilm-based wound care are just refocused aspects of wound bed preparation. But the key of that is frequent debridement to remove the biofilm colonies. And second, then, to use an effective microbicidal dressing after the debridement to prevent reformation. In addition, you need to frequently change the types of antimicrobial treatments you're using after sequential debridements because the bacteria adapt within the biofilms to different treatments.